Hey guys, so today you are gonna learn about the topic tetanus. Okay, tetanus. So now, which organism will cause tetanus? So organism that is that cause tetanus is Clostridium tetani. Okay, Clostridium tetani. So this is a anaerobic, obligate anaerobic. Okay, obligate anaerobic, and it is a gram-positive organism. So this organism, it will produce two types of virulence factors. So these virulence factors are, one is tetanolysin and another one is tetanospasmin. Okay, so this tetanolysin, it has no role in the pathogenesis of the tetanus. Okay, now this tetanospasmin, it is a neurotoxin. Okay, neurotoxin, all the manifestations that are produced is because of this tetanospasmin. Okay, now let's see the pathogenesis. Okay, so now this tetanospasmin is there, no? It is going to bind to the receptors of polycyallogamma. This tetanospasmin is going to bind to the receptors. These receptors are polycyallogangliosides, which are present on the motor nerve terminals. So these receptors are present on the motor nerve terminals. So after this binds, so there occurs an internalization. Okay, internalization. Now after this internalization, this toxin is carried to the GABA and glycine producing inhibitory neuron terminals. Okay, so these are transported to these inhibitory neuron terminals in an retrograde manner. Okay, so what this toxin is do is, this toxin prevents the release of these inhibitory neurotransmitters, and which will lead to spastic muscle contraction. Okay, this will lead to the spastic muscle contraction. So now let's see the mode of transmission. Okay, how it is transported. What is the mode of transport? First, it is to the injury. Any injury to the rusted objects, punctured wounds, any road traffic accidents like that, open wounds. Okay, injury. Another one is surgery. Okay, surgery done in aseptic conditions. Okay, surgery that is done in aseptic conditions. And in the neonates, how these neonates will occur? So neonates occur this infection due to unhygienic practices during the abortion or delivery. Okay, unhygienic practices during this abortion or the delivery. So clinical manifestations. So this clinical manifestations, and first we will see the incubation period. So the incubation period is 6 to 10 days. Okay, incubation period is 6 to 10 days. So now in clinical features, so the first symptom is trismus. Remember this very well, trismus or lock jaw. Okay, so what it will do, this causes the increased masseter tone. So this will occur because of the masseter muscle. Okay, masseter muscle tone is increased. Okay, now in the neonates, how they will present? Neonates are presented in the way there is feeding difficulty. Okay, feeding difficulty is seen in the neonates, and as the disease is progressing, there is painful muscle spasms. Okay, painful muscle spasms are seen, and later you can also see descending spastic paralysis. Descending spastic paralysis is seen in this patient. So now complications. What are the complications you can see is, one is rhesus sardonicus. What is rhesus sardonicus? It is because of the abnormal sustained spasms of the facial muscles. Okay, sustained spasms of facial muscle. Now another complication is, another complications is opisthotonous position. So what is this opisthotonous position? It will occur because of the contraction of the extensor muscles. Okay, remember, extensor muscle contraction. Now let's see the lab diagnosis. So lab diagnosis is like, first you need to take the specimen, means from the wounds, or you need to take the bits of the specimens Okay, now next do the gram staining. In the gram staining, you will see gram positive bacilli, okay, and in the positive bacilli, in the gram staining, you will see drumstick appearance. How? Like this. Drumstick appearance of the organism, okay. Now, another one is Robertson cooked meal broth. In the culture, you need to do Robertson cooked meat broth. So here it is, as this organism, it is a proteolytic, means as Clostridium tetani, it is a proteolytic. It will turn the meat particles black and produces foul odor, okay. This bacteria, it will turn the meat particles black plus foul smell is released, okay. That's it. So this is about the pathogenesis, laboratory diagnosis. Now in the prevention, so what you will do in the prevention, okay. So this prevention is done by active immunization. So this immunization, one is tetanus toxoid. So this tetanus, it is a commonly used, okay, this is commonly used tetanus toxoid. So this is a monovalent vaccine. This is a monovalent. What are the other combined vaccines are? One is DPT vaccine. DPT vaccine, this contains diphtheria, pertussis and the tetanus. Okay, another one is TD. It contains tetanus plus diphtheria toxoid. Okay, these two. Another one is pentavalent. Okay, pentavalent, this contains DPT, hepatitis B and HIV, Haemophilus influenza B. Okay, these are the immunization. Okay, where you will immunize? So this tetanus is an intramuscular. Okay, so this is an intramuscular. Where you will go is anterior lateral aspect of the thigh. Anterior lateral aspect of the thigh. Okay, and in the adults, you will given the deltoid. Okay, anterior aspect of the thigh in the children, and in the adults you will given the deltoid muscle. So this is all about tetanus. I hope you guys understood. So thank you.